Today we'll be talking about the urinary system. The urinary system is divided up into two parts. The first part has to do with the kidney and the nephron. The second part has to do with the function of the kidney and the excretory ducts. Urinary system, Part 1, the kidney and nephron. Part 1, we want to describe the structures of the kidney and distinguish between the cortical and medullary regions. We want to identify and differentiate different portions of the nephron, and we want to trace the blood flow through the kidney. That's what we do in Part 1. And here we can see the kidney, ureters, and urinary bladder, which are the main components of the urinary system. We've got the cortex on the outside and the medulla, and then you've got the minor calyx, major calyx, pelvis, ureters, and the urinary bladder. And the blood enters and exits as well as urine through the hilus region. And here we can see by the body whorls, the kidney, the hilus region in either one, ureters, urinary bladders, there's a prostate, urethra coming through the penis of the male. Of course, male and female have the urinary systems. Now, what's the function of a urinary system? The function is to maintain homeostasis. It rids the body of waste, that is urea, uric acid, creatinine, and salts, and it preserves the constancy of the extracellular fluid in composition, volume, and pH. So it conserves the constancy. It also has an endocrine function, secretes erythropoietin, which is important in red blood cell production. It also produces renin, which causes aldosterone release. So the functions are to rid the body of waste, to maintain extracellular fluids, and to have an endocrine function. And here we can see again the kidney, ureters, and the urinary bladder, blood flow going into where arteries and veins enter and out through the hilus region, cortex, medulla, minor calyx, major calyx, and the pelvis. And also here's a nephron, the main unit, functional unit, and structural unit of the kidney is a nephron. And you can see where the blood arteries interacts with the nephron at the glomerulus. And then from the glomerulus, you go to the proximal convoluted tubule, proximal straight tubule, loop of Henle, straight distal tubule through the collecting tubules, and we'll talk about those as we go through. Surrounding the tubule is the blood vessels. And here again we can see the cortex and medulla, the minor calyx, major calyx, and you can see the artery coming in through here, going through an arcuate artery that makes a little arch around through there, through this medulla, cortex, medulla, and the medullary cords that come in through there, cortex, and medulla, or the renal columns, where you have cortical material that goes between the medullary cords. And here we can see the capsule of the kidney. Capsule is adventitious type capsule. These are the tubules in through there. There's a capsule, no mesothelium on the surface. And then in the center here, we can see where fluid leaves out through, this is like transitional epithelium, that goes through in the minor calyx in through there. So the cortex, medulla, renal pelvis is right in through this region. This is the minor calyx of the renal pelvis. Here we see the cortex and medulla. Let's see medullary rays run up in the cortical material, as you see in through there, the arcuate arteries going in through there, and where it interacts with the nephron, the blood interacts at the glomerulus. So again, we can see the arcuate artery coming in through there. With a higher mag, you can see that artery sends out other arteries that project up toward the surface, and from those is where the glomeruli are attached. So if we follow the blood through all the way through, you start out with an artery, renal artery, and then a segmental artery, so it segments. And then you have the interlobular artery right in through here, interlobular. And then the arcuate artery that makes an arc over the pyramid, the medullary pyramid. And then coming up from there, you have the interlobular artery, and then from the interlobular artery that you see coming in through there, you have the afferent arteriole, and then then the glomerulus, and then the efferent arteriole, and then it can either go through the vasa recta down deep, or the peritubular capillaries, which are located around the nephron, and then from there it goes through back through the vein, which is the interlobular vein, the arcuate vein, the interlobar vein, and then the segmented vein, and then ultimately to the renal vein. And that's basically what we have here as the flow. As you can see, it goes through major vessels, minor vessels, eventually to the glomerulus, and then it makes its way back after it goes through either the peritubular capillaries, or the blood goes through the vasa rectae. And so here we can see a piece of kidney. This is the capsule in through there, cortex, medulla. These are medullary rays that you can see projecting up through there, which is the cortical material that projects up. It's the medullary material that projects up in the cortex. We can see those running through there, and there's a glomerulus in through there, medulla. And the ones next to the medulla are the juxtamedullary nephrons. So these are different than those in the cortex, as we'll see later on. So the function of the kidney is brought about by a certain mechanism. One is filtration. It filters out the things. It keeps the blood cells. It also is active in secretion, secretes things. Then they have passive diffusion, something go in and out, and then a selective absorption. So those are the mechanisms by which it occurs, and occurs, of course, in the glomerulus, to have the filtration to occur, and then in the other parts of the nephron is where you get these other components to occur. So as we said, the basic components of a nephron is the renal corpuscle, where it houses the glomerulus. Glomerulus is a part of the vascular supply that projects in through there, and from there it goes to the proximal tubules, loop of Henle, distal tubules, to the collecting tubules. And here, if we can see the glomerulus more easily, this is the corpuscle right in through there, and you see you have an afferent going in and efferent going out. You have some extra cells, some mesangial cells that are located in through there, and then you have endothelial cells lining the blood vessel, of course, and then you have the glomerular capillaries, as you see, which are the blood vessels. Then you have the podocytes. The podocytes are the cells through which the fluid goes after it gets out of the blood vessel to go into Bowman's space, and then from Bowman's space it goes out the proximal tubule. And so here we can see that the afferent, which is usually larger in caliber than the efferent arteriole, and then once it goes here, it goes to these peritubular capillaries located around the nephrons. The nephrons in gray, and you have the proximal tubules, and then the distal tubule, which comes back up next to the glomerulus to the vascular pole in through there, then go to the collecting ducts, and then ultimately out through the renal pelvis. A little bit about evolution of the kidney. It occurred, of course, as animals trying to escape predators. So when the jaws were developed and animals became predatory of other ones, there was reason to flee from those. And here we can see salt water in through here and here, and the darker water is brackish water. So as animals escaped from the sea, they went into the rivers with brackish water, and the first thing they found was that they had plenty of water, but not enough salt. And that's where the glomerulus was developed. Here we see the glomerulus developing, and it's associated with the mouth of the early nephron in the beginning, and the early nephron was probably just something that drained gametes, and then finally it was embedded in a wall, and we'll see in a minute how that occurs. But then as animals become amphibians and they walked on land again to escape the predators, they not only needed salt, but they needed water, and that's where we got the long loop of Henle to develop to preserve water. Some of the animals have returned to the sea. Some of them have lost their glomerulus. Others actually have glomeruli, as you see. So as you see, glomerulus was associated early on with the opening of this tube, and then later it was embedded in the wall of the tube as what we have today. And so here we can see where the blood vessel was embedded in a wall. So these cells here are the podocytes. These are the parietal cells, and through there. And so the podocytes are really part of the original wall. So these are the podocytes, which would be the same as what we just saw on the outside, the cells that are in Bowman's space, as we can see. Not only something happens to this end, but also something happens to the blood flow direction. Note that all the blood is coming from different directions in here, but all of a sudden here, the blood only comes from the efferent arteriole comes down into this region, into peritubular capillaries, and that made a portal arteriole. So whenever blood goes through two capillaries, 1, 2, that is a portal vessel. In fact, that's what a portal vessel is the vessel between two capillaries before the blood returns to the heart. And so if we see this, we can see the afferent arteriole coming in, bringing blood in, and then we have the efferent arteriole taken out beyond the glomerulus, coming to the peritubular capillaries, and you can see them here and here, and these are the peritubular capillaries. So the function of a portal system is you have, of course, two capillary networks. Here we see the first capillary network is in the glomerulus, and the second capillary network is in the peritubular capillaries. And what happens is that the function of a portal system is to have local change in blood composition, whereby the first capillary modifies the blood, so the blood is modified in the glomerulus, and then the second capillary network benefits from the change in composition from the cells. So the blood is modified in the glomerulus, and here in this case you have the cells of proteins stay back, you have a high osmotic pressure, and then that blood goes through here and it returns the fluid back to these capillaries because of a high osmotic pressure. First capillary network modifies the blood, the second capillary network uses that modification in a local situation. So here we see the arcuate artery coming in here, the afferent arteriole, the capillary, goes through the efferent, through the peritubular capillaries, or the vasa rectae. Now the vasa rectae are peritubular capillaries too, they're just located down deep in the medulla, and then finally to the arcuate artery and then out. But here we can see the glomerulus interacts with the Bowman's corpuscle, Bowman's space in through there, proximal tubule, loop of Henle, distal tubule, and then the collecting tubule, as you see. So we can see those, as we just said. Remember that the glomerulus is actually embedded into the wall of the nephron, efferent arterioles, peritubular capillaries. If we look more at the renal corpuscle, you can see that you have a capsule around a glomerular tuft, and you have the podocytes, which were part of the original wall, surrounding the capillaries, and the capillaries are fenestrated for maximum exchange to occur. Here you can see the glomerulus. These are the podocyte scheme of transmission electron microscopy, and here you can see the filtrate. This would be blood, red blood cells over here, and the filtrate on this side, which will include some glucose, amino acids, ions, many hormones, and things like that, some of which will be reabsorbed. So if this is your renal corpuscle, the glomerulus, podocytes is what we see, efferent arteriole. This is a distal tubule coming in through here, and is going out through the proximal tubule. So blood comes in, filters, and the filtrate goes out through the proximal tubule. So here we see where blood comes in, the so-called vascular pole versus the urinary pole, which is where you have a continuation of Bowman's space and the lumen of the proximal tubule. Again, we can see Bowman's space. These are podocytes right in through here that's projecting out into the lumen, and these are parietal epithelium on the outside. So here we can see the parietal epithelium on the outside, as I mentioned. It's actually part of the wall of the original nephron. And then you see the podocytes. Podocytes, again, part of the wall, and it's what the Bowman space is between the parietal epithelium and the podocytes. Mesangial cells and endothelial cells we can't see because they're covered. But the mesangial cells are the extra cells in through there, and the endothelial cells are the ones that line this capillary. So here we can see the endothelial cells lining this capillary in through here, are part of the glomerulus. These are podocytes in through there, and these are parietal cells, and the mesangial cells are the extra cells in through there. Mesangial cells are kind of like pericytes in blood vessels, but we can see that mesangial cells secrete erythropoietin, which is important for red blood cell production. So here we see the podocytes over the capillary tufts. At this slide 32, we can see a glomerulus. This is a vascular pole, urinary pole in through there. This is Bowman space, parietal epithelium, podocytes on the surface, endothelial cells lining these capillaries. This is a glomerulus, and this is a distal tubule. These are proximal tubules, and here the more eosinophilic ones, less eosinophilic ones are the ones that are the proximal tubules. Another Bowman space in through there. So this whole thing is a renal corpuscle, Bowman spaces between these two. Here we see proximal tubules here and here, and this is a glomerulus in through there and a distal tubule. The proximal tubules have a long distance between adjacent cells, as you see, where the distal tubules and nuclei are fairly close together. Proximal tubules have a brush border, only microvilli in the distal tubule. So here we can see the proximal tubules are very eosinophilic in through there. Distal tubules here. This is the vascular pole, distal tubule, Bowman space. Juxtaglomerular cells, these would be the JG cells that are located in through there, and there's mesangial cells in through there. Microvilli, the brush border of the proximal convoluted tubules. At the bottom of these tubules, the proximal tubules are infolded. They're infolded like this, and this kind of lines up the mitochondria. You see that in the proximal and distal tubule associated with pumping out electrolytes. On the apex, in addition to the infolding you see at the bottom, in the apex, you have the brush border. The brush border and a lot of apical cavity canaliculi, little projections where you can see in the next one, this is animal has been perfused with albumin, and it was able to get through, some of it was able to get through the glomerulus into the filtrate, and the proximal tubule, the brush border, trapped it, and to take in the protein, to reabsorb protein. However, a little bit of protein is considered normal. 0 to 8 milligrams per deciliter of protein in urine is considered normal. So you can't get it all, but it's important to return as much protein as possible. Here we see the kidney. This is the capsule in through there, no mesothelium, as I said. Proximal tubules, most of these tubules are proximal tubules, renal corpuscle. The intense eosinophilia that we see in the proximal tubules of the cytoplasm is partly due to the glycocalyx of the brush border, which stains intensely eosinophilic. Also due to the high density of membranes within the cells. So the cells don't have granules. What they have is a lot of membranes, mitochondria, infoldings, or brush border. All those things are lots of membrane proteins that we see, which are red. These membranes and a high density of proximal convoluted tubule in the cortex are responsible for the cortex absorbing most of the filtrate. In fact, we'll learn it's 80% of the filtrate from the renal corpuscle. So here's the glomerulus again, and this is the urinary pole. So this is the beginning of the proximal tubule. This is proximal tubule here, 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 and there. Maybe distal tubule right there, not sure. But then we have the collecting tubule. This is a distal convoluted tubule and a proximal convoluted tubule, and this is a collecting tubule here. Collecting tubule uses a bigger space, more clear cells. In the distal tubule, you have lots of mitochondria at the base and the enfolding of the plasma membrane, as we'll see in a minute. So where the afferent and efferent arteriole comes in and out, that's the vascular pole. And the parietal leaf, visceral leaf, which is the podocytes, and the filtrate coming in. Then you have a urinary pole. And so we see the vascular pole and the urinary pole where Bowman's space is emptying with the proximal tubule, where you can see the brush border of the proximal tubule. These are proximal tubules in through here, glomerulus, parietal epithelium, podocytes on the surface, parietal, podocytes, glomerulus, distal tubule, proximal tubule, proximal tubule, proximal tubule. Most of these are proximal tubules. All these guys are proximal tubules, a little bit of distal tubule in through there. This is the urinary pole, so this is the same as that. The urinary pole, and maybe the vascular pole is where it's attached up here. Bowman's space and the renal corpuscle, and these mostly are proximal convoluted tubules. And slide 33, which is staining for PAS, and the basement membrane of these cells are PAS positive. All epithelium sits on the basement membrane, so you can see it at the bottom of these duct cells. Also, you can see the pink that runs through there is the basal lamina between the endothelial cells and the podocytes as well. Distal tubule here, you can note you don't have a brush border that you have here with the proximal tubule. Distal, no brush border. You do have microvilli, but you have brush border. And what is staining here is the brush border that you receive, and it has a rich supply of glycocalyx covering the numerous microvilli in the brush border. So it's really sugars on the brush border that's staining carbohydrate rich. Here we see the podocytes. There's major and minor processes as they entirely cover the capillaries. The capillaries themselves are fenestrated. We see more podocytes, and these are sitting in Bowman's space. So the different components of the barrier. Here's podocyte. This would be Bowman's space. This is a capillary, so this is be the endothelial cell, and you can see it's fenestrated in through there. So you have fenestrations, as you can see them there. You can also see them right in through here as well. Also, the thick basement membrane, which is shared between the endothelial cells here, and the podocytes on the other side. And then finally, the filtration slits. So the filtration slits are minor processes that have finger-like process that projects over and covers the endothelial cell. Has a little membrane over there, and here we see the little slits. And those are the three things, and you can see those here. One is at the pores of the fenestrated capillaries. It's the basement membrane, which is shared by the endothelial cells and the podocytes. And then finally is the filtration slits. You see the little slits here. So this is blood, and this is filtrate in through there. Just for ballpark values, endothelial cells stop large proteins and cells. Basal lamina stops another amount, and then the filtration slits stops yet further. And that means some things are trapped in the basal lamina. That's where the mesangial cell migrates through and may be involved in cleaning up the basal lamina. Here we can see the histologic features of various of the components, as I mentioned, where they're located, and what their function is. So you can take a look at those. And here we can see the proximal tubules has a brush border, as I mentioned, infolded with lots of mitochondria in through there. Distal tubules, a lot of infolded microvilli, as opposed to a brush border that you have in a proximal tubule. Thin loop of Henle, and then the collecting tubules seem kind of washed out because they're kind of more passive, as opposed to these guys are really active and pumping. Proximal tubule, distal tubule, collecting tubule, and you have the peritubular capillaries and the vasa rectae that goes in through here. We can see that again. This is the thick portion of the loop of Henle. If you look right in through there, that's what that is. And the ascending going back up, distal, and the descending proximal tubule in through there, and then the thin component of the loop of Henle. So here we see the collecting tubules, more obvious. You can see distinct boundaries between adjacent cells. The proximal tubules are a big distance between adjacent nuclei, and then these are the thick portion of the loop of Henle and the thin portion of the loop of Henle, and then we see the capillaries. There's a host of capillaries in through there. Again, we can see that the loop of Henle varies in thickness, as you see in through there, and there, versus the capillaries. Capillaries have a thinner wall. You can see the blood in the capillaries in through there, blood capillaries versus the thin portion of the loop of Henle, and this is a collecting tubule. Now here we can see an electron microscopic view of a transition between the descending proximal tubule and the loop of Henle. It's descending portion of the loop of Henle. So you lose your brush border abruptly and become simple squamous cells. This is peritubular capillary located right in through there, and we can see those. So here's a transition on one side. It's proximal tubule on the other side. It's the thin portion of loop of Henle all the way down. And here we can see another transaction, another transition, another transition, another transition. Multiple transitions, and you can find that very frequently because there's such a long span of it. It's easy to cut them across. And what we're talking about is a transition between the proximal tubule and the thin portion of the loop of Henle. And we can see that again. So this is a transition right here. So this is what it looks like in the proximal tubule, and this is what it looks like in the descending portion of the loop of Henle. And we see another transition here and another transition here. These are distal tubules. And through here, 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 and here are distal tubules. And here we can see a transition of the ascending portion of the loop of Henle and the distal tubule. So these guys here, kind of bubbly looking cytoplasm, as you see, are the ascending portion of the loop of Henle, as opposed to these guys, which are smooth in through there, thick and smooth, as you see here, are the descending, ascending portion, and you can see a transition between that ascending portion of the loop of Henle and the distal tubule. And you can see another one right here. So this is the lumen of the ascending loop of Henle and the distal tubule. We can see another one. These are transitions you can see. This is the distal tubule, and this is the thin portion of the loop of Henle. So there's one transition. Here you can see another transition between these cells in through here, which are the thin portion, ascending portion of loop of Henle, and the distal tubule. And we're really talking about that between here and here, between the ascending portion and the distal tubule coming back up in through there. In addition to that, we might note that the mitochondria, due to the infolding of the plasma membrane at the base, the infoldings, it lines up the mitochondria, kind of like striated duct, for example. And that makes the striations that sometimes you can see. You can see the mitochondria very nicely lined up, and electron microscopic view confirming what we can visualize. So here we see the descending thin portion of the loop of Henle, as opposed to the ascending thin portion of the loop of Henle, which is continuous with the distal tubule. And here we can see the capillaries. See, the capillaries of the vasa recta because we're down low. So we see one here, one there, one there, one there. The wall of the capillaries are thinner. Endothelial cells are thinner than are the walls of the loop of Henle. So again, we can see collecting tubules, the thin loop of Henle, in through there and through. That's actually the thick and thin portion of the loop of Henle. Collecting tubules. Collecting tubules, you see, in through there usually gets real tall toward the end, and the distinct boundary between the adjacent cells, as you see here and here, and you can see the distinct boundaries, no interdigitation between adjacent cells, and the cytoplasm looks kind of washed out. And so you come down from the collecting tubules, and then ultimately they end there at the minor calyx, So cortex, medulla, collecting tubule ends down here. We want to thank the various books, magazines, websites that provide the original drawings, pictures that was modified for this presentation. That's the end of urinary system part 1, the kidney and nephron. Next we will have the urinary system part 2, which will be function and excretory ducts. Thank you.